Our next speaker is Dr. Yael Mosse. She has received her bachelor's degree at Smith College in Massachusetts and her MD at Sackler School of Medicine in Tel Aviv. She did her residency and postdoctoral fellowship at Children's Hospital of Philadelphia. She is Associate Professor of Pediatrics at the University of Pennsylvania, Perelman School of Medicine. She is a member of the steering committee of the Children's Oncology Group. She is chair and vice chair of several clinical trials within the COG, and she has authored and co-authored numerous articles and publications. Her team defined the mechanism of malignant transformation and has led to the routine genetic testing in the clinic, as well as the identification of ALK as a tractable therapeutic target. Her talk today is on the evolution of precision medicine. Dr. Yael Mosse.
Thanks very much, Pat. I really am very grateful for the invitation to be here. It's an honor for me, and to see a lot of incredibly familiar faces is very heartwarming and heart-wrenching as well. So I appreciate the opportunity to talk about my sort of vision of the evolution of personalized medicine and how it applies to neuroblastoma. And I thought it would be good to start with some definitions so we're all on the same page about how we define personalized medicine, precision medicine. And so really I think of it as individualization of patient care. And while it feels like a new concept, it really is something that's been around for a long time. Personalized genetic medicine is the practice of medicine that uses an individual's genetic profile to guide decisions made in regard to prevention, diagnosis, and treatment of disease. And then some people call it precision medicine, but it really is the same thing. So genetics is the study of genes that you're born with, whereas cancer genomics is the study of how these genes become disorganized, not in your constitutional DNA, but in the tumor itself. So it starts off as the DNA that then gets translated into RNA that then gets translated into protein, which is the actual target. So when we'll talk about, for example, ALK is the protein that's druggable. So when we talk about sequencing to understand what are the targets, we talk about sequencing at the DNA level. And that defines the mutations in the DNA that then lead to the abnormal protein drug targets. And the proteins are the molecules that really perform whether it's the different cellular mechanisms and signaling in your body, whether it's to cause increased growth of cells or cause arrest of growth of cells. So there's a lot of talk about sequencing, but it's important to decipher that when we talk about cancer, we talk about a disease of two genomes, the one that you're born with, the one that is your constitutional DNA that you get from your parents, and that is your germline. And this has really become very important as we think about pediatric cancers and susceptibility to pediatric cancers. So your germline is the genes that you inherit from your mother and your father. They're in every cell of your body. And you can inherit a bad gene or a mutated gene in your germline that predisposes you to the development of cancer. When we talk about the somatic genome, which is the other genome, that is really only used to refer to the genetic changes that occur in the cancer itself. And personalized medicine, especially in pediatric oncology now, really considers both the germline and the somatic genome. So our vision of personalized neuroblastoma care is that we want to provide monitoring for those children who are predisposed to developing disease. And it's really been our data suggests now that every child diagnosed with cancer should have genetic screening and genetic counseling. We think it's going to be very important to use this information to provide more precise prognostication. So if there's an ALK mutation, does that mean that your child has a better prognosis, a worse prognosis? But perhaps the ultimate goal is to use this information to provide more rational and precise therapies. We also have an opportunity to provide the ability to monitor and change therapy prior to clinical disease progression, and that's really where we're going in this field, is to be able to detect low-level cancer cells floating around in our blood before a patient's disease becomes resistant to our therapy. And then to be able to tailor therapy to an individual's ability to respond to a targeted therapy. We're trying to do that also in the realm of trying to figure out if a patient is going to have a very adverse reaction to our chemotherapy agents, and to be able to utilize that information to alter their therapy. So in reality, as I mentioned at the beginning, although it feels like precision medicine is really sort of this new era in cancer investigation, it's really not. And even in neuroblastoma, we've actually been personalizing medicine or thinking about personalized medicine for quite a long time. And when we discovered mutations in the germline in genes like FOX2B and ALK now, FOX2B was in 2004 and ALK was in 2008. And with this information, we are now providing genetic counseling to families. We've been using MYC-N as a prognostication factor for a very, very long time. And actually, our current therapies targeting GD2, so dinutuximab, targeting the norepinephrine transporter using MIBG, these are targeted therapies. This is precision medicine. And we've been doing this quite a long time in other diseases, especially in leukemia, where we monitor residual leukemia cells in the bone marrow to change therapy based on the presence or absence of those cells. And we now even have the ability to identify children who are more susceptible to really toxic side effects from our chemotherapy, and can adapt our therapy based on the identification of these changes. So wanted to highlight, before getting into some of the clinical trials that we've been developing in this era of more precise medicine, to highlight some of the obstacles and pitfalls and how we're addressing those. So genetic predisposition to neuroblastoma is very complex. It's not just all about ALK and FOX2B. There are, in fact, over 20 DNA variants that are known to contribute to neuroblastoma risk. And most of these are individually very weak, but a perfect storm of several of these is likely causal for individual children who develop neuroblastoma. And we're not yet in a place where we can identify these in individual patients, but we certainly are aiming to be there soon so that we can provide genetic counseling for families. And what do mutations in adult cancer predisposition genes mean for children? So as we are now doing all this sequencing, if in your child we find a mutation in a gene, BRCA2, that predisposes to breast cancer, what does that mean? And is this something that we should be thinking about now, that we should be acting upon now? So we are very focused in our efforts to identify mutations in genes that can be targeted therapeutically, but we then find also these incidental discoveries of mutations in genes that could cause other diseases later on, and how do we address that? And this is an evolving field, and the cancer genomes themselves, so not just the germline, but the cancer itself, is highly it's sort of this plastic system that is very, you can really stretch it. So it looks one way before you are treated with chemotherapy, and then after chemotherapy, our therapy induces a lot of mutations that then cause the cancer to evolve and become a whole different disease. So it may look like neuroblastoma under the microscope every time that it's biopsied, but the evolution of the cancer genes is become extremely complex. And the reality, as you all know, is that when a child unfortunately suffers a relapse, you don't need a tumor biopsy to diagnose that. And so, what is the prospect for benefit to offering that to families? So I'm going to talk a lot about ALK, because ALK has really become the only tractable therapeutic target in neuroblastoma. We discovered it in 2008 after almost 20 years of collecting the rare families where neuroblastoma is inherited. And as technology evolved, and as we collected a few more really key 3-generation families where neuroblastoma segregated, we discovered in the germline of these rare families that mutations in this gene ALK, which, let me see if I, so it's shown here in its stick figure. This is the cell surface. This is the surface of a cell. This is what sticks out on top of the cell, and this is what's inside the cell. And all of the mutations reside in this really key domain called the kinase domain. What's really interesting and makes this very different from other cancers where you hear about the ALK gene is that lung cancer, the mutations all happen where you don't have this extracellular domain anymore. There's nothing sticking out on the cell surface. So there's a cut here, and the ALK gene then is paired up with a number of other genes in the genome to form basically a new gene that causes cancer. But in neuroblastoma, we have the ALK gene that remains in its intact form. And the reason I mention that and emphasize that is because the fact that it's expressed on the cell surface here makes this a really good target for immunotherapies. And we're working very hard to develop antibodies and cell therapies to target ALK on the cell surface. The other reason this is relevant is while only a subset of kids have mutations here in this part of ALK, almost all neuroblastoma tumors have this part of ALK expressed on their cell surface. And so our efforts to develop antibody therapies targeting ALK on the cell surface will be relevant to probably 90% of kids with the high-risk form or the resistant form of neuroblastoma. Whereas the therapies targeting the mutant form of ALK are only relevant to those kids whose tumors have a point mutation or a genetic alteration within that portion inside the cell. So what we know is that at diagnosis, ALK is the most frequently mutated gene in neuroblastoma, and it's an oncogene, which means that these mutations turn the gene on, which gives us a really unique opportunity to work with pharmaceutical companies that have been developing drugs to turn the gene off in non-small cell lung cancer to apply that to neuroblastoma. So this is a collection of 206 neuroblastoma tumors at the time of diagnosis. And when we use very old sequencing methodologies, we find that at diagnosis, about 11% of children have a mutation in the ALK gene. But we now have much better technologies. They cost a lot more, but with these technologies, we can actually sequence the tumor very, very deeply, and we see that probably an additional at least 6-7% more have mutations in the ALK gene. And right now, we have not been able to bring these technologies to clinical care, but that's what we're working towards in our laboratory at CHOP, so that we can identify not just the 11% where there's an ALK mutation in every single cell in the tumor, but also the 7%, and perhaps even a greater frequency than this, who are likely to benefit from an ALK inhibition at diagnosis. And the reason I really highlight this is that you can see all these other genes. These are mutations, so MYC-N, P53, RAS. These are very infrequently mutated, but ALK mutations are likely much more frequent than we even appreciate right now. But perhaps what's really striking is what happens when we expose children to our very toxic therapies. So this is what, it's hard to see these plots, but this is a tumor, a collection of 23 tumors at diagnosis, and then this is those same 23 tumors when the children suffer a relapse. And this is kind of two ways of plotting the same thing, which is the mutation rate at diagnosis and then at relapse. And I think the sense I really want to give you is that upon exposure to our therapy, which we recognize is extremely toxic, the genome changes dramatically. So the cancer genome acquires a lot of new mutations, and many of these mutations are in the pathway of ALK and in this other very important cancer pathway called the MAP kinase pathway. So that when we look in tumors at relapse, this is just a study we did in collaboration with Foundation Medicine. Many kids have had their tumors biopsied, sent to Foundation Medicine for sequencing, and we were able to look back at all these kids with neuroblastoma, and what we see is that the frequency of ALK mutations at relapse is 20% compared to, it really ranges in the literature. It's between 7 and 9%, 7 and 11%. So this suggests to us that actually these are probably present at diagnosis, but not in every single cell. And then our therapy allows the ALK mutant cells to become they don't respond to our conventional therapy, our chemotherapy, and then they become the dominant resistant clones. So they become responsible for the relapse, and at relapse, we see a much higher rate of ALK mutations. And in fact, I'll show you new data that suggests that the frequency of ALK mutations at relapse is closer to 40%. So with these data and a lot of other data that I won't get into, but always happy to talk about, we felt that we now really have the rationale to talk with families about performing a biopsy at relapse, because at relapse, it's a very different disease. And we have an opportunity to really identify mutations in druggable pathways that maybe we really didn't understand at the time of diagnosis. So we now feel that we're in equipoise, that the potential benefit to a child from sequencing a relapsed neuroblastoma outweighs the potential risks of performing such a biopsy. And it's our recommendation that kids with relapsed neuroblastoma should be offered a biopsy, that families should be offered a biopsy, and that the tumor should be subjected to sequencing using the right technologies to obtain the most amount of information to guide therapy, so that every child has the opportunity to benefit from a therapy that is rational and precise for them, that could really differ from a therapy that would make sense for another child. So it's our vision and our expectation that sequencing of all newly diagnosed neuroblastomas and their matched germline will happen at diagnosis, so we can tell you at the beginning whether a child has mutation that's just in the neuroblastoma tumor or whether they have a predisposition to developing neuroblastoma. How does that affect the whole family, children in the family? And we're very hopeful that we will be moving into an era where we can perform liquid biopsies rather than actually having to do a biopsy of a bone metastatic lesion or a soft tissue lesion, that we can actually now capture these mutations by just doing a blood draw. And we're very much getting there. So some of the challenges in using this information to develop trials is that the presence of a mutation does not equate to the knowledge of an effective therapy. So an example is that this gene BRAF is an important driver in 50% of adults with malignant melanoma. And we know that there are several FDA-approved drugs now targeting BRAF that have dramatically improved the outcome for these patients. But this same gene can be mutated in colon cancer at the exact same position in your DNA. But those adults with colon cancer don't respond to the same drugs used in melanoma to turn off the exact same gene. So BRAF in melanoma is not the same thing as BRAF in colon cancer. And this has been exactly the story for ALK, that all ALK mutations are not created equal, and they're very different from ALK in lung cancer. And so this is where, the underlying biology is really important. And as a physician scientist, I don't think that there are nearly enough investigators doing the critical experiments to define the effective matches between a target and a drug. It's just much more complicated than a binary yes-no. And so this is where, we think that all of the work that happens in the laboratory is really directly relevant to each individual child. So what is preclinical data? And this really gets to how do you generate the robust experiments in the laboratory that support providing a personalized recommendation for an individual child. So these are the experiments that provide the proof of concept that ultimately give us the approval by the FDA and our local regulatory bodies to open a clinical trial. And it's typically a series of pretty extensive studies in our Petri dishes, but perhaps much more importantly, in our animal models. And the point I really want to make here is that many of the more unique animal models that we've been able to generate over the past couple of years have been from children at the time of death, where the families have allowed us to draw a large volume of blood and establish a model that represents their child's disease, that allows us to test our drugs and our hypotheses in a way that we think is much more authentic than doing so in a Petri dish. And when you generate robust preclinical data in the right models and you have the right controls, then, only then really, do you have an opportunity to translate this into a clinical trial. So many years ago, we started working with Novartis Pharmaceuticals, and they have a very comprehensive panel of different drugs being developed for turning off different cancer signaling pathways. And we did a screen with them where we sent them 30 neuroblastoma models, and they took a bunch of their drugs and they crossed them to each other. And what we did is we divided up the models we had in those that had an ALK mutation and those that did not have an ALK mutation. And we identified some combinations that only made sense in the ALK mutant models and some combinations that only made sense in the models where ALK was not a player. And these are the data that we then took to models in our animal system. And so I'll highlight this particular model. This model was, we now no longer call it COG N426. This is now called Felix. This is a model that we generated from a little boy who came to see us from the United Kingdom in our early days of our understanding of ALK, where his tumor was sequenced in the UK after he suffered a relapse and showed an ALK mutation, and the family relocated to Philadelphia. But at that time, what I knew from my laboratory work is that this mutation was completely resistant to the only ALK inhibitor we had, which was the ALK inhibitor he came to Philadelphia for, which was crizotinib. The first ALK inhibitor, the first FDA-approved ALK inhibitor for lung cancer, I knew would do absolutely nothing in the context of this mutation. And ultimately, when he died, his parents allowed us to draw a large volume of blood, and we were able to establish this model that that's now named after him. And we were able to show that when we combine a pretty standard ALK inhibitor called ceritinib, it's Novartis' ALK inhibitor, it functions similar to Pfizer's ALK inhibitor, crizotinib, when you just use it by itself. When you combine it with a drug called ribociclib, which causes cells to arrest in the cell cycle, it causes massive cell death. And so the two agents together are very effective at shrinking the tumors completely. Whereas each agent by itself, shown here in all sort of these different arms, different doses of the drugs used by themselves, completely ineffective. So this provided a lot of the rationale for a trial that we've had open at CHOP, and it's been only open at CHOP because of the complexity of this trial. It took us 5 different contracts to get this open, and to be honest, it took us almost 3 years to really get this open to patient accrual. It opened in August of 2016, and I will say that we're working very hard to have this open at 3 additional sites in the US within the next 6 months. And this is what we think is the first precision medicine trial for children with relapsed neuroblastoma, where at the beginning, we talk about a biopsy based on the scan today, and we review that scan with our radiologists, our interventional radiologists, our pathologists, our experts in nuclear medicine, and really think about what is the best lesion to biopsy, what represents the child's disease today. And then the child, if the parents consent, has an outpatient-based biopsy. The majority of these are bone lesions. And the child goes home that same day. And we then, in collaboration with Foundation Medicine, send that tissue off for sequencing. The turnaround time is 10 days. And then we get a result. And then we meet again with families and discuss what available therapies we have for children with relapsed neuroblastoma. And right now, the trial has had 3 different treatment arms. One is for children who have ALK mutations, where we're combining that ALK inhibitor with the cell cycle inhibitor. So any child with an ALK mutation gets assigned to this therapy. Patients whose tumors have mutations in another signaling pathway, the MAP kinase pathway, which is mutually exclusive from mutations in ALK. We have some really compelling data that when you combine a MEK inhibitor, trametinib, with a cell cycle inhibitor, that there's nice activity. And so actually this treatment group is just opening. And the reason that these trials have been such a challenge for us to open, but have also really provided us a unique opportunity, is that these are just in phase 1 in adults as well. So in adult lung cancer with ALK mutations, they are also just still in phase 1 with this combination. Adults with MAP kinase pathway alterations, this combination is also in phase 1. It's actually been to the credit of Novartis to provide us with an opportunity to follow right behind them rather than to wait until they got to phase 2 or phase 3. But we've also had to be cautious with our dose escalation. And then the third group is for kids whose tumors don't have a mutation in the MAP kinase pathway or ALK, where a very important gene that is a master regulator called P53, as long as that gene has not acquired new mutations, this is a gene, this is a drug that allows for that pathway to be reactivated and really perform the functions that it was meant to perform its functions as a master tumor suppressor. And this is an interesting treatment group because what we've learned about kids with neuroblastoma is that as we expose them, unfortunately, to more and more of our very toxic therapies, the tumors can lend themselves to acquiring mutations in this P53 gene. And we unfortunately learned in the laboratory that if the P53 gene has a mutation, this drug is completely ineffective. But that's actually important information because you really wouldn't want your child to get this drug. It's a complete waste of time if the P53 gene is mutated. So this is something that, again, is really important information in thinking about precise therapies. So right now, these are the treatment arms that we have, but this is serving as an umbrella trial where our goal is to bring to more combination therapies is really our goal. We're really not interested in single agent therapies. Our goal with this drug here is to combine it with other interesting drugs. And that's where we're moving forward. So what you can see here is sort of the flow diagram of how this therapy works, where we get a next generation sequencing report, and then depending on the mutation, a patient is assigned to one of 3 different treatment groups. One of the interesting aspects of this trial, which we think makes it a much more objective trial, is it's all based on the work we did in the laboratory. So there's no tumor board that meets. There's no sort of consensus. We always say consensus is not science. So it's a very objective assignment to treatment based on laboratory work that's built into the protocol. And as part of this trial, we've had a really unique opportunity to collect tumors from patients not only at the time that they've relapsed and we have the opportunity to do a biopsy and to learn from that, but parents provide us with the permission to get all the archival tissue on their child. And now we're doing all this serial sequencing to learn what happens over time and over the exposure to chemotherapy. And this is our experience. This is very new data. First 27 kids enrolled on our NEPENTHE trial and what the sequencing results have showed us. And it's a relatively unbiased group of patients referred to us for screening for this therapy, for this clinical trial. And what we're finding here is that 37% of these tumors have an ALK mutation. So remember I said at diagnosis, it's between 7 and 11%. With better technologies at diagnosis, we think it's maybe more like 18%. Then we saw it was 20% at relapse, and now we're seeing it's really closer to 40% at relapse. So this is, makes a compelling argument, at least for a biopsy at relapse. The disease at that time is a very different disease than it is at diagnosis. So I think many of you who have had an interest in ALK, whether it's from just an interest in neuroblastoma biology or your own personal experience, know that the first generation ALK inhibitor that we took from very initial discovery in 2008. Now it's going to be the next phase 3 trial in the Children's Oncology Group just opened 2 days ago. And that trial is the first phase 3 trial that we're going to perform sequencing of the tumors at diagnosis, and all children whose tumors are found to have an ALK mutation will be non-randomly assigned to getting crizotinib, the first ALK inhibitor that was developed, in combination with our standard therapy. So the standard induction, our 2 transplants, our immunotherapy, with the goal that if ALK is really important in the growth of neuroblastoma, that targeting it with crizotinib is going to make a big difference in our ability to cure these kids. But we know that crizotinib is not good enough. It's actually pretty good in combination with chemotherapy. But as a single agent, it's not good enough, and probably no drug as a single agent is going to be good enough. But we've been working very hard to identify better ALK inhibitors. We've tested every ALK inhibitor that's either FDA approved or in early phase clinical trials for adults with non-small cell lung cancer. And we found that this drug lorlatinib, which has, if you can look at its chemical structure, looks very different than crizotinib, is really far superior. It's a much more potent ALK inhibitor and at much lower doses. And again, we tested it in our animal models. And I'll bring you back to Felix here. So these are mice who have neuroblastoma tumors engrafted in them. When the neuroblastoma tumor gets to a certain size, 10 mice get randomized to just getting like a placebo. 10 Felix mice got crizotinib. See, this is why we didn't recommend crizotinib for Felix, because it really does nothing. And then the mice who got the lorlatinib really have dramatic and sustained tumor regressions here. And we see that in these other models that have mutations that don't respond to crizotinib. Now, interestingly, here's a model that has a mutation at this residue 1275. And our early observations were that crizotinib was a good drug for this mutation. But our early work only carried out our animal experiments to 4 weeks. So what happened was, in blue, so we treated the mice. By 2 weeks, the tumors were gone. And we carried out the experiment to about 4 weeks, and then we would stop. But in this experiment, when we tested it against lorlatinib, we carried out the experiment. And you can see that even though crizotinib looks good for a couple of weeks, despite the fact that we still treat the mice every day, the tumors become resistant at about 3.5, 4 weeks. Whereas with lorlatinib, it appears to be a much better drug. So we have a big effort to try to understand, why all of that is. But more importantly, we were able to relatively expedite the translating these findings into a phase 1 trial. And the reason I say relatively is because we designed the phase 1 trial so that if we see the activity that we hope to see, that I would like to say we expect to see based on our laboratory work, that this could serve as data that we could bring to the FDA for a new drug application. And so the trial took us a little bit longer to develop than we had hoped, but it did open to patient enrollment in September 1st, 2017. So just a few months ago. It's been open for about 6 or 7 months. And it's extremely well-tolerated drug. You gain weight. Kids are happy. It's very different than crizotinib. It's a whole new experience. It has very different specificity for ALK, so it's just extremely well tolerated. And we initially thought, based on our work in the laboratory, that we didn't have to get to really high doses. For crizotinib, we had to get to really, really high doses to even begin to think that we would see responses. And so we designed the study to only have 3 dose levels, where the first dose level would be about 80% of the adult recommended phase 2 dose. Dose level 2 is about equivalent to the adult dose, and then dose level 3 is about 25% above the adult dose. We've actually completed all 3 dose levels and see no toxicities at all. It's extremely well tolerated, and this is what we found. And these are all new data that I'm sharing with you because I think it's really important to be very transparent about the status of ongoing clinical trials. And for me, what's really important is doing the work in the laboratory in parallel to these clinical trials. What are we learning? How can it feed into the clinical trial? And so, some of our colleagues at Pfizer felt like, okay, we're done with the 3 dose levels. We're going to call dose level 3 the highest dose, and we're going to just move forward now to expanding. And I said, let's start to look at the pharmacokinetic data. So parents give us the opportunity to collect blood at serial time points from their children. And what we learned is that the levels that we were achieving were the same as in the adults with non-small cell lung cancer. But remember, the biology in neuroblastoma is very different. And so it became very clear to me when I went back to our work in the laboratory that, and we published this in a supplement to our paper, that these are the curves I showed you. But to achieve these doses, we need some higher exposures. So right now we're at about this dose. And what happens here is that the tumors are stable, but they don't go away. And that is not good enough. And so the trial is currently suspended because we're amending it to add more dose levels. And I feel very fortunate because I work with a company that's not risk adverse or minimally risk adverse, and who understands the biology and who understands the need in the population and the importance of really finding the right dose for the right population. So this is the reason why this trial is currently not open to patient enrollment, but will be open soon. So really, in conclusion, I think it's important to be very wary of clinical trials that don't have robust laboratory-based proof of concept. And you can get this information from physicians. You can just ask for it. That's what we're here for. And while it's important to explore all options, just to be wary of personalized therapy recommendations that sound too good to be true. With that, I will just end by saying that when we talk about precision medicine, it all seems like it's all at the level of the DNA, the genomics. But as we move forward, we know that precision medicine really means just precisely identifying therapies that make sense for an individual child, and it goes way beyond genomics. So immunotherapies now are really where there's a significant effort to develop precision therapies. Thank you for your time, and happy to take questions either here or later.
Okay. Thank you. Does anyone have any questions? No? Okay. Yeah, if you could go up to the microphone.
Hello, Dr. Mosse. I'm Brandon from San Antonio. I have a question is, what are the odds of not finding any targetable mutations? Or having a mutation, while it's found, it has no relevancy into how you can approach it, such as what I've understood as ATRX deletion.
Yeah.
And the third part of the question is there's different institutions that are doing sequencing. Some are more robust and some are thinner. Foundation One, I think, was our original ones that we've done, and MTRC does a different panel. And you do, how do those compare, and how do y'all work with each other to make sure they're comprehensive?
Yeah, those are all very important questions, and thank you for highlighting these challenges. So the first is, drugging the undruggable. And the reality is that the majority of the time, there aren't going to be precisely druggable mutations. And this is where a huge effort is ongoing now, and why the laboratory work is so integral is really understanding. So ATRX, is an example that you gave, and there were a couple talks at this meeting about how can we drug that pathway. Are there drugs developing that target other nodes in that pathway that are relevant to ATRX tumors, or ATRX-driven tumors? And there is some definite, new data now that's compelling. But that is a big challenge, and this is why we really need to, in parallel to these obvious matches that we have, is to really be working towards when there aren't matches. So working with pharmaceutical companies, having a larger portfolio, but really understanding the underlying biology. As far as the different platforms, we're now in a place where the majority of platforms will give you the important information that will allow you to make recommendations for treatment. The nuances are all very important to consider. So for example, Foundation Medicine has developed a way now to calculate what's called tumor mutation burden, which we think is a marker of understanding whether a patient is going to respond to immunotherapy. So this new checkpoint blockade era that we have. And so that's not something that all companies are doing, but this is something that now Foundation Medicine has shared with other companies so that they can utilize that. But I think the most important thing is to talk with your primary team about these platforms and which one makes sense. And the liquid biopsies now are extremely powerful too.
Okay. Anyone else? Okay.
Hello, my name is Saeed Salihat from Bay Area. First question is easy. I wanted to know, with liquid biopsy, who are you working with and how far along are you? And second question is, what if the relapse, let's say, is in the bone? Do you still do a biopsy, and how accurate is it?
So for the liquid biopsies, again, we've been working with Foundation Medicine. We found them to have a really robust research and development group. It's actually being implemented in our lorlatinib trial in the NANT, so every patient has a liquid biopsy done at the time that they're enrolled and throughout their therapy. And we're already learning a lot from that information. It's a panel of 60 genes, mostly devised towards adult cancers, but they've actually optimized their platform based on our interest in pediatric oncology. And your second question, remind me again? Yeah, so the bone biopsies. Yeah, so the data that I showed you for our sequencing efforts in the 27 kids, 25 of those were bone biopsies. And only 2 did we not get what we call lesional tissue, and that's because what we got was what was mature ganglioneuroblastoma as opposed to undifferentiated neuroblastoma. So there are now very precise ways to superimpose MRI on a bone lesion with an MIBG and put the needle right in the tumor and get data. And I think several parents here who I have worked with closely can really speak to that. Thank you very much.
Thank you, Dr. Mosse.